Hello everyone, my name is Dinko Rekic, and on behalf of my colleagues Jane, Katrina, and Bengt, it is my pleasure to talk to you about the use of model-informed drug development and how it enabled 6 months acceleration of a phase 2 study. This example comes from the hyperlipidemia space. Hyperlipidemia, or high cholesterol, is one of the key risk factors for cardiovascular disease, which is the #1 cause of death in the world. There is a well-known causative link between cholesterol and major cardiovascular endpoints, and LDL cholesterol is recognized as a surrogate endpoint by the FDA. Despite the availability of high-intensity statins, there is still an unmet medical need in this field, as 50 to 80% of patients do not reach their LDL-C treatment goals. A new class of injectable drugs emerged in 2015. Monoclonal antibodies against PCSK9 lower LDL-C by approximately 60%, but they come with a cumbersome dosing regimen. In 2021, inclisiran, a small interfering RNA dosed every 6 months, was under FDA review with approximately 50% LDL-C reduction. Our goal was to develop a PCSK9 inhibitor based on the antisense oligonucleotide platform with a convenient dosing regimen and a best-in-class LDL-C lowering profile of 70%. In order to accelerate and de-risk the development program, we embraced model-informed drug development at all levels in the project. In our single ascending dose study in healthy volunteers, we observed a great dose response on PCSK9 reaching more than 90% inhibition after a single dose. This data was modeled with a KPD model. The same model was used to conduct clinical trial simulations to predict what level of PCSK9 inhibition we would observe after repeated monthly dosing. In order to understand what kind of LDL-C reduction that would translate to, we digitalized all available data in the PCSK9 class from the literature, shown here as gray circles. We also conducted simulation based on published models from the Repot and inclisiran teams. Both data and models pointed to a single relationship between PCSK9 and LDL-C reduction, showing that in order to reach our target of 70% LDL-C reduction, we needed to inhibit PCSK9 by 90%. This meant that already after a single dose, we had enough confidence in our best-in-class profile to invest in an accelerated development program with significant front-loading. At this point in time, we approached the FDA as part of the MIDD Pilot Program. We wanted to show the agency how well PCSK9 correlates with LDL, and that we can use it for setting doses for phase 2 study based on the data from the single ascending dose study and limited safety data from the ongoing MAD cohorts. FDA agreed that we could start the phase 2 study as soon as sufficient safety data was available, and we were able to save approximately 6 months in time in clinical development. Shortly after the MIDD meeting, our data from our first multiple ascending dose cohort in patients on top of statin was available. Using our KPD model developed on the single ascending dose PCSK9 data and the historical LDL-C relationship, we made predictions on how LDL-C lowering would look in this cohort. These predictions were made in January 2020, shown here as teal bands. The observed data, shown as circles, were remarkably well predicted. This gave us confidence in our approach of using historical PCSK9 LDL-C relationship and further increased confidence in the program to deliver the best-in-class profile. At this point in time, we had sufficient data to make an informed choice about what doses we want to test in phase 2. The PCSK9 KPD model was updated based on the new data, and we simulated PCSK9 reduction at steady state for monthly doses up to hundreds of milligrams per month. Based on these simulations, we estimated that a dose of 50 milligrams would be sufficient to reduce PCSK9 by 90%. We also conducted probability of success calculations based on the simulations, showing that a 50 milligram dose had over 80% probability of reaching 90% inhibition in 50% of the subjects. The sample size needed to select the right dose was evaluated using an approach similar to the one proposed by Smith et al., where the estimated confidence intervals for LDL-C reduction were compared to a target value of 70% and a lower reference value of 65%. So assuming a true effect of 70% for the high dose and a 65% for the low dose, We showed that the correct dose was selected in 95% of the simulations with a sample size of 20 subjects per arm or more. Based on these analyses that heavily relied on modeling, we were able to select doses for phase 2 study and make sure the study is properly sized for dose selection. In conclusion, model-informed drug development had and continues to have an enormous impact on the development of a new PCSK9 inhibitor. Based on the single ascending dose study, we were able to gain early confidence in the efficacy of the compound that enabled significant front-loading and investment with a reduced risk. The FDA MIDD program enabled us to start a phase 2 study prior to completion of the MAD study, leading to a 6-month time gain. And finally, we increased efficiency in our phase 2 design without compromising the probability of success. This led to cost reductions and increase in speed. At the time of this recording, the next steps for this program is phase 3 dose selection based on the readout of the phase 2 study. Finally, I would like to thank and acknowledge my colleagues who have made significant contributions to this work. Also, I would like to thank you for listening today.